So to begin with, I thought maybe I'd try to conjure up the origins of modern astronomy and microscopy, since that's sort of those are the topics this evening. So imagine, if you will, we're about 1608, and the telescope is invented somewhere in the Netherlands. And within about a year, Galileo Galilei gets wind of it in Padua. And he has expertise in lens making, and he crafts fine lenses. And he's really the first person to turn this telescope on the heavens. And you can find in his notebooks, imagine, drawings of the surface of the moon with craters. And he draws them as craters. He recognizes them as craters. He looks at Jupiter and sees star-like objects near Jupiter. And as the days go by, they change position. And he infers that they're actually circling the planet of Jupiter. He gazes on the Milky Way and resolves them into stars. I mean, these are just, it's hard to put yourself in his shoes, but maybe that will give us a bit of a sense of the dawn of astronomy. Microscopy follows some decades later, 1673, in Delft, Antony van Leeuwenhoek, he's a draper, and he wants to make some kind of a magnifier to look at the fibers in his cloth. And he does that. He's developed some skill in a secret way of making tiny spherical lenses. But he's also an amateur scientist, and he's the first person to look and identify bacteria and other single-celled animal-like creatures. So these are the very beginnings, and the instruments that Dimitar and Adam use are the almost unimaginable descendants. They're not simple meter-and-a-half-long telescopes or microscopes that you can hold in a hand. They look almost nothing like that. I think they'll have some images of what these instruments are like today. But they are, in some sense, the descendants of these two great pioneers. Since the super topic, in a way, is light, as we heard, it might also be interesting to note that in 1611, Galileo traveled to Rome with something that, again, somebody else had really discovered, which was barium sulfide. And barium sulfide was a kind of black crystal that in a dark room glowed. It gave off light. And this at the time meant that light was not the same thing as heat. It was not always associated with heat. It wasn't always something to do with transparent materials. There was something special. He recognized that light was something separate. And actually, some of the techniques that Adam Cohen talks about are cousins of this. This is phosphorescence, and he will talk about fluorescence, but it's a related process that was not understood until more than 300 years after that demonstration. So about the speakers this evening. Dimitar Sasselov was born in Sofia, Bulgaria, and you can imagine him maybe as a child amateur astronomer. I don't know if this is really true, trying to recapitulate Galileo's discoveries. He had telescopes. He looked at the moon. He looked at the heavens. He went on to study physics. He got degrees in physics, a PhD in physics at the University of Sofia. But this was the late 80s, and following the revolutions of the late 80s, he came to Toronto and got, wouldn't you know it, a second PhD, this time in astronomy at the University of Toronto. And he is now the Phillips Professor of Astronomy here at Harvard and Director of the Origins of Life Initiative. Adam Cohen, as a boy, built a scanning electrochemical microscope that hung from bungee cords in his bedroom, and with it he won the Westinghouse Talent Search Prize. He came to Harvard, earned degrees in physics and chemistry, and went off to the University of Cambridge to get a PhD in physics. Actually, one of his advisors is Mahadevan, who's now a professor here in applied mathematics. And not content with a PhD in theoretical physics from the University of Cambridge, he went to Stanford University to get a second PhD, also in physics, this time experimental physics, with W.E. Morner, who a year ago won a Nobel Prize in chemistry. And Adam is a professor of chemistry and chemical biology and of physics here at Harvard. And I think that Dimitar is speaking to us first. So, Dimitar.
Thank you, Kenny, and good evening to everyone. Well, the macro world, that's the discipline of astronomy, science of the big universe. We use the tools of physics to study the universe. Essentially, that's the definition of the science of astronomy. So, astronomy and light. What comes to mind, to your mind, when you think about astronomy and light? Well, what comes to my mind, and I think to yours too, is a very dark night. One like this, a dark place somewhere in Vermont or further away in the mountains, where you can see the Milky Way, the stars. So what is astronomy? Astronomy is 100% about the light, not about the darkness. So it is those glimmering, shining dots of light that we are after in astronomy. And if you train a big telescope, like the Hubble Space Telescope, on those stars, they shine really bright. And this is one of the deep images from the Hubble Space Telescope, one of the premier, still very actively used scientific instruments for all of humankind's astronomy, for that matter. So this is a very typical telescope, though by today's standards on the ground, it's a smallish one. But it does beautiful work, and it has used the power of light in many different colors to tell us about how stars form on the tips of those fingers or pillars. These are young star forming regions, or behind the dark areas in this Horsehead Nebula, as it is known, in the Orion constellation of Orion. It also has told us about the end of the life cycles of stars, or dying stars, as we would actually call them among ourselves in astronomy. These are stars which have gone through their cycle, similar to the sun, and they are beautiful but ephemeral shells of hot gas dispersing and being just for a short period of time illuminated by the very hot core of the dying star in the middle. And they actually show those colors because they fluoresce because of the UV light that is emitted in the middle. We also, in astronomy, come closer to home and use the power of our tools that study light to study the planets, like Saturn in this beautiful photograph in contre-jour from the sun behind it. And all the way to the edge of what we can see, which in this case is the deep field, the so-called Hubble deep field, where we see galaxies, those little specks that you see there, with the exception of maybe 2 or 3 on this picture. This one is a star, but everything else are very, very distant galaxies. And some of them are as far back as we could see with our current technology, almost at the edge of what we would call the visible universe and the age of galaxies formation. So I want you to contemplate for a minute this image and what it means, because that is one property of light which is very important to astronomy and which defines astronomy as a science to a great extent. What you actually see here with these different specks of light is a picture of a world that was. What you see here is an image of things, galaxies in this case, that existed billions of years ago. Some of them 2 to 4 billion years ago, some of them almost 10 billion years ago. The reason is because of the limited speed of light. Light, as fast and instantaneous as it may appear to us here, the tiny creatures that live on the surface of this planet, actually moves at a limited speed. And when it encounters the amazingly large scales of the universe, it actually takes a long time. Sometimes it's frustrating to scientists as well that they have to wait hours before they get those images, those photos of the surface of Pluto, most recently this summer. In this case, we have to wait billions of years. But turn this around and think about what an amazing tool this is. This is the only science in which you're actually witnessing the past of your subject matter as opposed to reconstructing it. I'm not asking you here to imagine that this is what the universe looked like 5 billion years ago, or galaxies like that look. I'm not asking you to trust me that I reconstructed it properly. It is what it is. This is actually, you're seeing the light, the images of those galaxies as they were 5 billion years ago, in some cases even older. So in astronomy, this is a very important and helpful thing. We actually do not need to reconstruct the past. We see it. And that's because light takes time to get to us. And this is one very simple diagram which shows how today's mapping of the past of the universe can be put in very precise sense, not only of distance, but also of time. And so the objects that we observe at larger distances also tell us what this kind of structures, galaxies of stars, were like a long time ago. And so then by studying many of them at different distances, you see their evolution, their development with time. And this in particular is the entire currently understood history, looking back, of the observable universe from a very early age, where inflation is yet to be understood and confirmed by experiments. But anything which starts with the afterglow light pattern at 375,000 years after that initial moment is actual data. It's not made up or reconstructed or theoretical. It is data. And so that's one thing about the history that we can study with astronomy. The other thing is the beautiful colors. Of course, you probably know in today's age of Photoshop and very good computer software, graphics software, you can do whatever miracles you want with your images. And it is true that those Hubble images that I showed you, and this particular one, another dying star here, we call them planetary nebula, but they are really the envelopes of dying stars. It is enhanced. The colors are enhanced, but they actually have a very specific meaning. And in fact, they're close to reality. The red-colored areas are predominantly hydrogen gas, which is emitting in that part of light, which appears red to us. The greener and bluer colors are oxygen, nitrogen, other gases. So in other words, when we talk about light, it is very important to remember, and I know you know this, that we only see a very tiny part of a very large spectrum, and we call it the spectrum, of different electromagnetic waves, of which light is part, very tiny part, of those electromagnetic waves, which is about the wavelength about half a micron. That's what our own eyes are adapted biologically to see. And we don't see infrared light, although we feel it as heat. We certainly don't see radio waves or microwaves. And thankfully, we don't see X-ray or gamma rays, which are very energetic. Ultraviolet, again, we feel, usually after a few hours as a sunburn. But that's the important point here, is electromagnetic phenomenon, those waves which are due to electromagnetism, the forces of electricity and magnetism, are what light is in that particular small area of the spectrum, which is around half a micron and which we call visible light, and where all the colors of the beautiful rainbow are actually squished together. The entire beauty of that world that we experience here on the surface of the planet and looking at the stars is only in that part of the electromagnetic spectrum, but the universe has all of those waves propagating through it and producing different amounts. Well, let's start with what is closer to home and what we understand, which is the visible spectrum. We use the visible spectrum to detect and characterize the planets in our own solar system, but particularly, most recently, planets around other stars. And that was the big investment by NASA in the last 5-6 years of the Kepler Space Telescope, which discovered a number of systems like this, planetary systems like Kepler 62. We use the light from the star and the colors to determine the temperature and size of the star itself. And then we look at the light to discover those 5 planets as they pass, in this case, in front of the star, eclipse it, and produce what we call transits. 2 of those planets, number 4, which you see now, and number 5, are in the so-called habitable zone, that is far enough from the star that they are not too hot and water can be in liquid form, although in number 5, Kepler 62f, it's a little bit colder because it's further out, more like Mars, while number 4 is more like Earth and Venus. So these 2 planets are of particular interest to us because they are similar to the Earth, and potentially we could look for signatures of life on them. So this is one of the big projects that is underfoot right now, and you must have heard about it, but it is true that a lot of our community in astronomy is now involved in both discovering but also characterizing those planets and really gearing up to look for what we call biosignatures, ways to detect the existence of biosphere similar to the microbial biosphere that has persisted on planet Earth for almost 4 billion years, as we know from the geological record. So how do you detect or search for life on a very distant planet where you cannot visit and sample that green stuff, which is this cyanobacteria that produced so much of the early history evidence for life on our planet Earth? Well, you use light again. The same tool which astronomy uses for everything. You go and observe the products of those microbes. And in some particular cases, like with the cyanobacteria, that's a very reactive gas which shouldn't be there geochemically. It's not getting out of volcanoes or out of the oceans or the continents. You have to have a living system to sustain a large number of oxygen gas. So how would you see the oxygen gas on such a distant planet with a telescope? Well, again, you have to go to the spectrum of light and say, well, what can I use here from the light that comes from those planets that can tell me about this existence? Well, in that little part here in the visible and infrared, which is the rainbow, there's a very fine structure which our human eyes, unfortunately, don't have the resolution to see. These are fingerprints of the individual atoms and molecules in a gas which emits or reflects the light that we see as a rainbow. We call them spectral lines. We cannot see them, and that's why we have to, with our own eyes, and that's why we have to attach to our telescopes very sophisticated instrumentation which does allow us to see those fine fingerprints of different atoms and molecules. We call them spectrographs, and the one that Harvard built together with the Geneva Observatory and is in the Canary Islands is right now working and doing some of this work on the mountain there. So what do we get? We get something which looks like that, where the individual molecules in the atmosphere, in this case, theoretically predicted for the same two planets, Kepler-62, E and F, would be detectable in that little visible part of the spectrum, which is here, and some of the infrared, which is the rest. So you see CO2, you see water, the stuff which we expect to see on those planets, and we know and we see when we look at the atmosphere of the Earth. But on top of that, we see things like ozone and free oxygen, which shouldn't be there unless there is life. So we call this remote sensing. And remote sensing has been a very successful way of studying things with the properties of light. I like this slide a lot. This is the World Soccer Cup in 2007 in Germany. And they have this little telescope here, which does exactly that same trick, which is what we astronomers want to do to detect life on exoplanets. Except in this case, they're only interested in alcohol. Guess what happened here? Somebody had too many beers. And you know, they want to make sure that the fans of the soccer club didn't get into trouble. So probably somebody went there and tapped on their shoulder. But that's how they did it, with this little telescope here. So we have the same problem or interesting things to solve here. We have planets which reflect light. They don't produce the light themselves. Well, the light goes through their atmospheres in transmission and in reflection. And so then we can see those gases. And we already have been able to do that. No, we haven't detected life on other planets, but we see the gases, so we know how to do it. And moreover, we can even do a little bit better because this is reflection. As you know from the summer is still here, and you still probably put your sunglasses today. You know that when sunlight reflects over a surface, it glares very unpleasantly. That's why we wear those sunglasses. That's because life, in addition to having a spectrum at different wavelengths, is also a wave which is simultaneously electric and magnetic fields. In other words, it has an orientation. And that orientation can be controlled with optical filters, but is also something which occurs naturally under reflection or in transmission. And we, again, in the future, plan to use that with the big telescopes of the future to detect those. And you would say, well, what would it take to do this? Well, it would take bigger telescopes. So for the big world, you also need big telescopes. Like, I assume Adam would need big microscopes. We need bigger telescopes. So Harvard is a partner in this giant, as it's called now, Magellan Telescope, coming up in 5 to 6 years in Chile. NASA is already building and almost finished the successor to the Hubble Space Telescope, which is the so-called James Webb Space Telescope. This is a prototype shown in Texas in 2013. And it's so big that you have to fold the mirror in the rocket so that you can take it to space. And of course, we also thinking about what if there are a lot of planets which have life on them. We'll know that probably in about 10 to 20 years. At least we'll have some statistical estimates. Microbial life, of course. how often does this microbial life develop into complex life? And is it possible that out there in the galaxy, there are a lot of signals of similarly developed complex life, which are simply the noise of their existence and their use of electromagnetic spectrum transmission and reception in order to do whatever they are doing, just like we do here on Earth. So you probably heard in July this year that there is a new push in SETI, the search for extraterrestrial intelligence, to get more telescope time and new technology, like the one you see here, which is called SETI at Home, which is essentially big data analysis with very large computers and new algorithms, to try again to see this time, not necessarily if anybody is sending us messages, but whether we actually hear or detect chatter or radars or similar electromagnetic equipment on any of the nearby exoplanets. So this is done in the radio waves. And as you can see, we use and are planning to use all the properties of light to advance our field. And now I'll let Adam tell you about the micro world. Adam.
Great. Thank you all for coming. It's really great to be here. So I'm going to pick up where Dimitar left off, starting with the planet Earth. Earth is full of beautiful colors, and many of the most beautiful colors come from life. It's full of magnificent colors. And these colors in life come from the genes, out of which contain instructions for all of the proteins that we're made of. And some of these proteins have colors on their own. Others are involved in assembling or synthesizing other molecules whose electronic structures are tuned so that they resonate with particular wavelengths of light and then have particular colors that we can see. Here's an example of one such creature. This is a jellyfish. Over the last few years, decades, people have learned how to take the genes from a creature like this to pull out the DNA, which contains within it the instructions for the proteins which are giving this jellyfish its beautiful color. You can take this genetic material and put it into a virus. And then you can take this virus and use it to transfer the genetic material to an embryo. And then you can grow up that embryo, and you can end up with a fluorescent monkey. And this is true, and this is obviously great because we all would really like to have fluorescent monkeys. This tool has been transformative for biology because it lets us associate these optical signals either with a whole creature or with different pieces of a creature. So we can use genetic tricks to target the expression, the activity of the gene, to just one cell or even one substructure within a cell, and thereby to light up things that we want to see. One area where this has been particularly useful is in the area of neuroscience. So if you put your hand on your head, your fingers are within 1 centimeter of the most amazing piece of matter in the universe. You can take your hand off now. We have about 86 billion neurons in our brain, and these neurons are connected to each other, each with about 10,000 synaptic connections to the others. And so we'd like to see these neurons. Here's a picture from a group that I'm working with. These are neurons from a rat. These are actually not inside of a brain. These are sensory neurons, which are being grown in a dish. And a gene from a jellyfish has been transferred to these neurons, and they're lit up so that we can see the cell bodies. Those are the meatballs, and the axons, those are the spaghetti that connect those meatballs. Okay? And these tools have been fantastic for trying to trace the connections within, for instance, neural circuits. But you can see here there's a challenge. If I asked you which cell is this particular piece of spaghetti here connected to, it would be almost impossible to trace because it's just too much of a jumble. And so Over time, scientists have taken genes which come from creatures of different colors, red, blue, green, pink, yellow, and developed a whole palette of different fluorescent proteins. These come from different corals and sea anemones, some eels, many different brightly colored creatures found in the world. And then it's possible to, again, using genetic tricks, to have different cells turn on different combinations of these genes. This is work that was done here at Harvard by Jeff Lichtman and Josh Saines's labs. And so here's an example now of a real slice of brain, now in living color, where each cell has a different combination of fluorescent proteins turned on. And so you can see, you can now track these connections in ways that wouldn't be possible if this were all monochrome. Here's another image from the same technique showing the cell bodies here, and then these are the dendrites coming out. This technique is fittingly called brainbow for labeling all the different colors. Now, these are beautiful images, and they help us discern the structure of these neural circuits. But they don't tell us what the cells are doing. It's a static picture. A topic that my lab has been interested in for the last few years is the question of how do we animate these images? How do we see the activity of the neurons? So I'll tell you a brief story that starts here. This is a view of the Dead Sea between Israel and Jordan. The water here is very salty. There's a single-celled microorganism that lives in the water here. It's called Halorubrum sodomense. And this creature produces a little protein which sits in the membrane of the creature. And this protein absorbs sunlight, and it uses that solar energy to generate a little electric current across the membrane. This electric current establishes a voltage, which this creature then uses to power its metabolic machinery. Okay? So you can think of this protein like the world's smallest photovoltaic device. It absorbs sunlight and generates electricity. Structurally, it's similar to the rhodopsin proteins in our eyes, which we use for vision, which also absorb sunlight, undergo a photochemical transformation, which leads to a signal in our eyes, which eventually signals up to our brains. So here, there are these amazing proteins which can absorb sunlight and convert it into electricity. So a few years ago, I said to myself, I wonder if we can run these things in reverse. Instead of having sunlight come in and an electrical energy come out, can we use an electrical impulse to generate some optical signal that we can see? Okay, so instead of light in and voltage out, we want voltage in and light out. Now, why is this interesting? Well, in our brains, the activity of our brains is encoded within tiny little electrical spikes in the neurons in our brain. So every thought and feeling and experience that we have is represented by little electrical spikes in our neurons that are about 1/10 of a volt high and about 1/1000th of a second long. That's all there is. So everything we experience is represented that way. And it would be really nice if we could see these electrical spikes. And so I thought maybe we could take a protein from this Dead Sea creature and put it into a neuron and then see the activity of that neuron as flashes of light. Okay? So now I'm going to skip several years of really hard work and tell you that we found a way to do this. Okay? So we started with neurons from a rat. Turns out you can buy rat brains online. There's a company called brainbits.com where you can mail order rat brains live FedEx overnight to your doorstep. And we mushed up these neurons and we grew them in a dish. And then we put the gene for this protein from the Dead Sea creature into a virus, and we infected the neuron with a virus. And here's a picture of a rat neuron. And you can't see it, but there's a little electrode stuck in the back of the cell. And I'll show you a movie now at 1,000 frames a second of what this neuron looks like when we trigger it to fire with this electrode. We have to image so fast because these events are very brief, only 1/1000th of a second. And so this is what a neuron looks like when it fires. So we can actually see the cell light up. Great. Now I want to talk a little bit about some of the changes in technology which are allowing us to do this kind of work. So traditionally, a microscope was a device which conveyed light from a sample to an observer's eyes, and then the observer would maybe write some notes or make a drawing of what he or she saw in the microscope. Microscopes today are nothing like this. Here's a photo looking down on one of the microscopes in my lab. This is obviously a home-built microscope. Each one of these little things is either a mirror or a lens. This is about 3 feet by 5 feet. And you might notice a few things about this microscope. The first is that there are no eyepieces. We don't ever even bother looking in our microscopes. And the reasons are that the signals we're trying to detect last 1/1000th of a second, far too fast for our eyes to see. And the light we're trying to detect is actually in the near infrared part of the spectrum. So even if it were slow enough, our eyes aren't sensitive there. We don't have any ability to resolve this light. So instead, we use cameras. This is a fancy camera, which is very sensitive and can run very, very fast. And then over here, just off the screen, is a huge amount of computational power. There has really been a revolution in our ability to analyze and interpret microscopic data because of the increases in computing power. This camera generates about 1 gigabyte of data per second as it's running. Now, there's one other thing I want to point out on this optical table, and that's this. This is a bank of lasers. Each one here is a different color, which we use for stimulating the sample. Until recently, people thought of microscopes as devices for passive observation of the sample. But if you think about life in the world, life responds to sunlight in a huge variety of ways. We can see, plants will grow toward the sun, plants photosynthesize, bacteria swim toward the sun. And so life has within it many proteins which respond actively to illumination. And nowadays, people often use microscopes in reverse. You use the microscope to convey patterns of light onto the sample in order to activate different chemical reactions or to control the activity of different genes and to actually get in there and push and pull on stuff inside the sample. And so we really think of a microscope as this two-way street now where we can use light to get a hook on the sample, and then we can use light to see what it did in response. So I'll show you some examples of this new approach of using light to control samples. So here's an example. This is a little single-celled alga. It's called Chlamydomonas. It likes to swim towards the sun. It senses the sun by having a little light-gated ion channel in its membrane. This is another protein that's structurally similar to the ones which we use for vision. The way this works is it absorbs blue light from the sun. That opens a little hole in the membrane. Some ions flow through, and that can change the activity of the flagella on this creature. So over the last 10 years or so, people have taken the gene for this protein and put it into some worms and mice and many other species, and thereby gained optical control over the activity of neurons in these other species. So here I'll show you some examples. This is work of Andy Leifer, who was a grad student here. This is a little worm, tiny one. It's about 1 millimeter long. And he's tracking it, and he's illuminating it with a little pulse of light right here around its midsection. And there's a particular spot right here where there's a single nerve which is responsible for controlling the muscle which makes this worm lay eggs. Okay? And so this worm is crawling along, and then at a certain point, the light will hit that egg-laying muscle. And there you see it. It starts to pop out eggs. Okay? And then when the light goes somewhere else, the egg laying stops. Okay, so you can use the light to trigger this thing to lay an egg. Here I'll show you another example. For the non-biologists in the audience, this is a mouse. And my friend Feng Zhang has injected a virus containing the gene for the same protein into the motor cortex of this mouse. So to control its locomotion. And so just to orient you, here this mouse, it's in a bucket. And you can't quite see it, but there's a little optical fiber going into its head. And I'll show you a movie of what happens when he shines a blue light into the brain of this mouse. So the mouse is exploring the bucket, and then at a certain point, the blue light will go on, and the mouse starts running in circles. And it'll keep running in circles as long as that blue light is on. And then at a certain point, the blue light will go off, and the mouse will return to its previous behavior. Okay? Here I'll show you another example where the same virus, oops, hang on, let me just pause this, has been injected into a different part of the mouse's brain, into a part of the brain responsible for controlling aggression. And so here's this mouse, it's in its cage, and here's a little inflated rubber glove. And you'll see when the light turns on, the mouse attacks the glove. Okay. And then at a certain point, the light will turn off, and the mouse will stop. And then the light will turn on again, and the mouse will attack the glove again. Great. So why is this interesting and important? Well, these kinds of experiments have allowed scientists to use light to dissect the roles of different circuits in the brain, different collections of neurons, in a huge variety of behaviors. So not just locomotion and aggression, but feeding, sleep, pup rearing, mating, social interactions. And what has emerged from this is the, to me, somewhat surprising observation that activation of relatively small numbers of neurons in defined circuits in the brain can lead to these seemingly hugely complex behaviors that these animals undergo. Okay, so the last thing I'll tell you about is some work from my lab where we've combined this ability to use light to trigger cells, neurons to fire, with this tool that we developed where we can record the firing of neurons with light. So we use blue light to make the neuron fire, and we use red light and this near-infrared fluorescence to look at the response. So here we're using the microscope in both directions. A neat thing about using light to stimulate a cell is that you can pattern the light. You can direct it in different directions and control where it goes in different patterns of space and time in order to activate the cell in different ways. So in this example, we've hacked a video projector. And we use the video projector to project the pattern of light onto the cell. Okay? So we're running this microscope in reverse. And so a projector is just like the one that's displaying this image. So this projector has about 1 million pixels in it. And so each one of those pixels now gets mapped to a spot in the microscope, in the field of view of the microscope, and corresponds to a stimulation in a corresponding spot in the sample. So here we're just stimulating the center of the cell. And in this movie, we've developed computational tricks to map the propagation, in this case, at 100,000 frames a second, to map the propagation of the electrical impulse. So we're stimulating here, and this is what we see. So you can really see in detail how these impulses propagate through the cell. So we're using light to stimulate, we're using light to record, and if you like, the impulse itself is an electromagnetic impulse propagating through the neuron, which is the substance of our own thoughts. So I have a company, a startup company, which is using this technology now to explore different neurodegenerative diseases. So as one example, this is a patient with ALS, a degenerative condition of motor neurons. And working with my colleague, Kevin Egan, who's in the stem cell department here, also a co-founder of the company, we've been making neurons derived from patients who have diseases like this, and then using these tools to characterize them and to study the differences in firing between neurons derived from patients and neurons derived from healthy people. And so these are some human motor neurons where we can now see their activity and try to use this to understand what has gone wrong in a disease like this. Great. So I want to end just by saying one word about where these tools come from that we're using. This voltage reporter that we work with comes from the Dead Sea here. The ion channel comes, in this case, it's from a freshwater alga, which lives in a pond in the south of England. It's fused to another fluorescent protein derived from a coral, and the voltage reporter is fused to a fluorescent protein derived from a jellyfish. And it's just incredible to me that we can take all of these colors, which came from different pieces of life, and then stick them together to come up with new functions which have never been selected for in the history of evolution. And the only reason we can do this is that basic scientists have been studying these seemingly useless creatures for decades, wading through the muck. All right. So anyways, I want to just thank the people who actually did the work in my lab. We're a brightly colored group. And with that, I think we'll turn it back over to Kenny.
Okay. I think maybe what we'll do is go straight to the audience. I mean, we were going to have a bit of a conversation, and I was going to seed it with some questions. But maybe looks like some people will be filtering out, and then after the room is settled down again, we'll open it to questions. And there are microphones up there, and if you can easily get to those microphones, please do. But if you can't, just stand up and shout out the question, and we'll reinterpret it or something like that. So we'll do it that way. Maybe just for a minute while people are filtering out, I will ask each of you to speak for a minute or two about the future, coming decades, where you see this sort of macroscopy and microscopy going. Let's say you pick a time scale, but be bold. Maybe go out to 50 years or something like that. And what do you think we'll be discovering?
Well, in astronomy, we have to plan those things about that long because it takes NASA or anybody, for that matter, to build those big telescopes. And so a lot of what is coming up is exploring further the invisible to the human eye part of the spectrum of light, particularly in the radio, which already started with a very big telescope, which is going to be built in the next 10 years to an even bigger radio telescope. Also, a lot of work is being done in the gamma ray range. And of course, closer to home, a lot of us want to see really more detail about what is on those exoplanets that look like the Earth. So that's where a lot of
your guess about intelligent life elsewhere in the universe? What's your guess?
I'm agnostic about it.
Agnostic. Oh.
Yes. Which means that I don't know. I have no clue. But I think ask me in 10 years, and I'll tell you why. I think it is important to do it in stages, to try to answer this question by finding out first how many planets are like the Earth. We already know that they're more than we thought. Second, how often, when the conditions are just like those on planet Earth, you actually have microbial biospheres. Those we can detect remotely. We know how to do it. And only then, once we know the answer to that question, statistically, I can basically hedge my bets on technology. But I think looking for the chatter, the radio signals back and forth towards the nearest stars with planets is worthwhile doing. Although the chances are nowhere close to 50-50 or even 1 in a million, it's worth trying. And I support that.
And the Fermi paradox, where is everybody? You know, if they're...
Well, maybe they're here, but we just don't know it. Many people think that we biological creatures are very inefficient when it comes to interstellar travel. We are too big, we carry too much excess water, and our brains can be much smaller than what they are. So they're probably right somewhere in the solar system. Well, this is jokingly so, but remember also our universe is very young. I basically showed you the entire history of the universe is a mere 13.8 billion years. In geological terms, that's nothing. So maybe we are the first out there.
And on the small scale or brains, whichever direction you want to...
Yeah. Well, I think there are still many really hard challenges in using light to study biology, to study small things. One area has been that, well, until very recently, the ability to resolve small objects was limited by the wavelength of light. Basically, it was hard to see things much smaller than the wavelengths we were using. And unfortunately, those wavelengths are, they're smaller than a mammalian cell, but only about 10-fold smaller, and about the size of a bacterium, or maybe half the size of a bacterium, and way bigger than most of the molecules we're interested in. And so much of the structure in biology had, until recently, been invisible. Now, over the last few years, people have developed a variety of techniques, super resolution microscopies, for doing better. But I would say there's still a lot of room to grow there. It would be really great if we could look at proteins or do optical measurements on proteins and see how the individual atoms are configured. I think that's one of the unsolved sort of holy grail problems, and there's no reason why it can't be solved. It's just we haven't been clever enough yet. Another one is to image, to see light through strongly scattering tissues. So the brain, unfortunately, is not transparent, and this is a big problem for trying to image what's going on inside of the brain. It's milky. But photons follow straightforward rules, and so it's conceivable that there might be ways to invert all of that scattering that happens and to find ways to see through the milk and to see the activity inside of a living brain. A dream which has not been realized is to have something like a mouse and to tickle its tail or to give it a stimulus and to watch the electrical impulses propagating through all the neurons in its brain as it responds. So I think that's another area where there will be huge advances. And then the third area, I think, will be on the development of new molecular tools to transduce light into different kinds of biological signals. So to use light to turn on and off different genes, to activate different signaling pathways in cells. And this could be extremely powerful for understanding things like embryonic development. Because with light, you can go in at particular times in particular places and deliver a very precise perturbation, and then you can see how that perturbation then manifests as a function of time and space in the developing embryo. And that's something which has not really been tractable with conventional chemical or biochemical techniques, where whatever you do takes a long time to apply and is spread over the whole embryo.
All right, let's throw it open to questions from anybody there. Okay, here's one.
So I want to ask Dimitar a difficult question. So can you offer some comments and personal thoughts on two issues? First, the varying speed of light, that is, at the origin of the universe, the speed of light could have been very different from that of the light now. And one of the purposes for that thought was to sidestep the inflation that you shown in the diagram. And second is the multiverse. Well, of course, both issues are highly speculative, but do you have any personal thoughts on these issues and comments? Thank you.
Thank you. Actually, the first question is quite a good one. It's not very difficult. It is true that when we talk about the speed of light, we always have to specify where this light, these electromagnetic waves are propagating. It's well known that light propagates slightly slower in thicker medium, like in glass, or even thicker than that. So we already know that when you talk about the very early universe, when the state of the universe is to be very dense and very hot, before the afterglow, which we observe directly, there was a lot of scattering, and indeed that whole concept has to be treated differently. However, I would say that this is the part of the story which we are still working on, and for which we don't have direct data or direct images or direct measurements. We only have direct measurements of the pattern of the sky, the universe, at the so-called cosmic microwave background, or the afterglow as it was in the picture. And what defines that pattern is that from there on, that is from that moment on, light can propagate at the speed of light in vacuum. So the rest of the story, actually, the ages are quite precise because the speed of light then is well known. And we see that pan out in the data. I didn't quite catch the second question.
Multiverse. The multiverse.
Oh, the multiverse. Well, the multiverse it's a long story. Basically, I'm not a big fan of it with my physics background, because essentially the reason for the multiverse hypothesis has to do with our understanding or lack of understanding of the basic structure of space-time, and particularly the structure below the currently known scales of the elementary particles. String theory is a very good effort to resolve that, but it has its own problems, which then result in some of the motivation for the multiverse hypothesis. And because these are two areas of physics and astrophysics which have no or very little, I would say no, experimental backing, it's kind of speculation on top of speculation. So that's as much as I like to talk about the multiverse.
So we're stuck with the one universe we have, I think. Somebody else? Other questions?
Here's a question.
Yes, please.
What about glia? This has to do with the neuron story and light. I mean, there are more glia than neurons in the brain.
Okay.
And do they have interesting voltage?
That's an interesting question. So the question is about the electrical properties of glia, which are also very important in brain function. And this is an area which has not been studied in enough detail yet to know for certain. It turns out to be very hard to measure electrical signals in glia. It's hard to get electrodes into them. And these glia also have, many of them have very long, thin processes sticking out from them, tentacles. And whether there is electrical signaling in those processes, it's simply not known. This is something that my lab has been interested in looking into, and I think there's actually a few people who are looking at this. Probably within the next year or two, we'll have a much better idea about this. We know that there are other kinds of signaling that happen in glia. So we know, for instance, that calcium signaling is very important in glia.
Somebody else? Yes, please.
So I believe that particular example of making this green fluorescent monkey was to demonstrate the capability to introduce genes from one species into the monkey and to get it to grow up and to show that that gene was then in every cell of the body. So the applications are then towards studying the role of other genes in either the whole monkey or in particular cells within the monkey. If I remember correctly, and I'm not 100% sure I remember this, is that in that instance, there was another gene which was either introduced or knocked out in the monkey, which had this green fluorescent protein associated with it. And so that fluorescence was a marker that the other perturbation to the monkey had also worked. And it was for the purpose of studying a disease in the monkey which had a genetic component. I don't remember what the disease was.
Are you going to trademark the fluorescent monkey? Because it seems that you have an opportunity to create a small fortune in creating a need that children should have orange pussycats.
Well, so I should be very clear. The fluorescent monkey was not my work. It was the work of somebody else. But you can buy at the pet store fish, zebrafish. These are small fish, about an inch long, which have different fluorescent protein genes introduced into them. And they come in all the colors of the rainbow. Yeah. Yeah, Ed.
I have a question for this guy.
Thank you. I'll repeat the question, so for those of you in the back. The question was in that particular picture, which is the Hubble Deep Field, where you see a very large number of galaxies of different distances and hence different ages. How do we know that this particular galaxy, say one of them, is at a particular age? And this is what took astronomers almost about 100 years to figure out. Harlow Shapley here at the Harvard College Observatory was famous for starting that hole, and Henrietta Leavitt, who designed one of the first methods for determining distances between galaxies. And so it has been a very long process in the astronomy community to bootstrap, that is to start with distances within our own galaxy, then to the nearest galaxies to us, to the satellites, then to the nearest galaxies, the Andromeda galaxy, which you can see by eye, and then all the way to those very distant ones which we measured with Hubble. In fact, one of the motivations to build the Hubble telescope was to fix the so-called cosmic distance scale. So it will take probably a whole course to describe that epic scientific work, but basically, we use this bootstrapping technique, starting here at home. Even the distance between the Earth and the Sun matters in that way, and slowly extrapolate all the way to those very distant galaxies. So this allows us, and has allowed us, to measure the distances to all of them. And then because the line of sight between them and us is usually clear, that was the previous question, with knowing precisely the speed of light in vacuum, we know how long ago that image left the source and is now registered by us. So we know some of these galaxies are 11 billion years old.
Have they done a regression picture of our Milky Way to see what it would look like if you mapped everything back to those at the same moment?
Yes, yes.
It looks significant.
It's very difficult to do a good job on that, but there are colleagues both in our department and in Toronto, in several places around the world who do this. They do it with large computer simulations where they put as many of the stars in the galaxy as possible and then run the movie backwards, as you would say. The difficulty in that is that we know that currently there are small satellite galaxies which are merging or falling into the Milky Way. And so we know how to regress that and see them leave. But we don't know how many others merged with the Milky Way in the past 10 billion years. And so to some extent, once you get that far back, because you don't have this information, the film is, necessarily only arbitrarily correct to some point. But it looks very similar to today's Milky Way, a little bit smaller.
That wasn't quite my question. I say, when I look up at the Milky Way, I see stars at all different ages.
Yeah.
Where their light comes from. What I was wondering was, what would the Milky Way look like, the image, if it was all at the same moment?
Like the shape of the Milky Way?
To some extent, you're asking a philosophical question which Einstein first encountered, simultaneity. We humans are used to instantaneous communication and being there in the instant. We call it the experience of the instant, the now. There is no such thing in physics or in the universe because most of the interactions, that is the forces and hence the information between what we call ordinary matter, the elementary particles from which we are made, atoms and molecules, is done with electromagnetic waves, light. And it's done at the speed of light. And so all these interactions take time. Even now, when you're looking at me, you're actually seeing me in the past, nanoseconds, or maybe less than that, but still. So that becomes a real difference in the solar system and beyond. And so that is the world in which we live. We just, in our heads, in our brains, we want it to be instantaneous. We want the now to exist because we exist in the now. But that's more philosophy than physics, I guess.
One final question, maybe, and then we'll wrap it up.
This is a question for Adam, but a comment for both of you. Just wow, and keep it going on minds a little bit. Tonight,
Good. So the question was, what are the differences between sick neurons and healthy ones, and how do we get these neurons in the first place?
And what do you,
Yeah. What are we looking for?
yeah.
What are we looking for? So let me answer the second part first. One of the big challenges in neuroscience and in the development of treatments for diseases of the nervous system has been the inaccessibility of human neurons. We don't do brain biopsies on people, and the reason is that the neurons in general don't regrow. And over the last few years, people have found a workaround, which is based on the fact that most cells in our body are pretty much genetically identical. And the only thing that distinguishes skin from bone, from neurons, from muscle, is which of the genes are there. But every cell has the seeds for the whole body. And so
Which of the genes are turned on?
which of the genes are turned on. And so people have figured out how to take some skin or blood from anybody and to reprogram these cells so that they go backward through embryonic development into a stem cell-like state, and then to take them forward down a different developmental pathway and to turn them into, say, neurons, which you can grow in a dish. And those neurons, they'll wire up and they'll start to fire. And they're genetically identical pretty much to the neurons in the brain or spinal cord of the person who gave the skin. And so for the first time, you can have a person sitting there. You can do this for anybody in the room. You can talk to the person, you can look at them, and then you can look at their neurons in the dish and see how they respond. Now, for each disease of the nervous system, it's a different story. There are many, many ways to break the nervous system. In the particular case of ALS, this is work that is in collaboration with Kevin Egan's lab, and he really started this and drove this before I got involved in this. His lab, in collaboration with Clifford Wolf at the medical school, observed differences in the excitability of the neurons from ALS patients with mutations associated with ALS from control neurons. And these differences correlated with clinical observations about the excitability of the motor neurons in these patients. And these differences in excitability then pointed to a particular channel, a particular gene in these neurons, which seemed to be hypoactive in the ALS neurons. And so then on the basis of that then suggested there was already a drug which was approved for treating epilepsy, which was an activator of this particular ion channel. And so then they started testing that drug in these patient-derived neurons in the dish. And on the basis of those experiments, GSK has actually just started a clinical trial of this drug for ALS. And so that's the kind of story that we're trying to now reiterate with many other diseases of the nervous system.
All right. Can you guys stick around for a couple minutes? Sure. So I think we're going to wrap it up there. Dimitar and Adam will stick around for a couple of minutes. If you have burning questions, you can come on down and gather around. But let's thank both Dimitar and Adam first.